All right, so thank you so much for the kind introduction. Again, I'm Tom Zajdel. I'm a graduate student in the University of California, Berkeley, and I'd like to share with you our ideas about how we might start to integrate actual living bacterial cells as front ends for sensing devices. So this is part of a larger effort in collaboration with Lawrence Berkeley National Lab, where we're actually trying to integrate living cells onto microrobotics platforms and use them for microactuation and microsensing. So this talk is kind of split into two halves. There's a fair bit of biology that we'll need to get through in order to really start to understand what happens when we apply our sensor to measuring the proton-motive force in E. coli. So I'd like to start with our vision. So here's an E. coli. This is an SEM. And they're about 2 microns long, but they're packed with all kinds of really interesting solutions to sensing problems and to microactuation problems. Billions of years of evolution have allowed E. coli to sense things in its environment and swim towards them and away from them. And so I really like to think of Escherichia coli or E. coli as the nanoscale multi-tool. So the ones we use in the lab are not harmful to humans, but they have all these really, you can do all sorts of genetic manipulation and start to take advantage of their sensing mechanisms. So I see a lot of opportunity that's really untapped. They can sense complex biomolecules, they can synthesize complex biomolecules and do interesting bioprocessing. They also can generate electrochemical potentials and store energy across their membranes. So our dream is really to use them as this front end for a sensor, for a sensing device. But in order to do that, we really need to understand what's going on inside. So what I'm going to talk about is what our target is, what we tried to sense in order to really start to understand what some of the dynamics are inside the E. coli. So our target is the proton-motive force. And this is an electrochemical potential that's set up across this membrane. So if we cut a cross-section in the shape of this rectangle, we end up noticing that bacterial cells are separated by their outside environment by 2 membranes. And the membrane of real importance is this phospholipid membrane, the inner membrane. And it's electrically an insulator, so there's a very high membrane resistivity. There's also a small capacitance that allows charge separation to take place. So you have a separation of protons from the outer membrane to the inner membrane, and they can't normally travel through the inner membrane because the inner membrane is nonpolar and you can't have ions travel through it. However, if you put in certain biomolecules that allow the proton current to flow, all of a sudden that potential, which is usually on the order of 100 to 200 millivolts, will allow protons to travel through. And you can harness that energy to do useful work, like chemical work, is it forming ATP? Or you can actually import molecules up a concentration gradient. Or very interesting to us is you can do mechanical work, like rotate the nanomotor, the flagellar motor. So the proton-motive force is really important for lots of different applications in the cell. So we really want to understand some of the dynamics of it. So the proton-motive force is our target, what we decided to measure. Now, in order to better understand some of what we're sensing, it's worth talking about a little bit how the cell generates the proton-motive force. It takes energy to separate that charge. And the way it does this is through its central metabolism. So it breaks down sugars like glucose and turns them into carbon dioxide and takes the electrons it gets from breaking the bond and moves it down an energy gradient that it uses to pump protons across its membrane. And that's how you actually generate the membrane potential. And then finally, when the electrons are mostly out of energy, oxygen picks them up and becomes the electron sink, and they become water. So you can think of this as sort of this redox tower where the electrons start at glucose, which has a reduction potential of minus 0.343 volts, and the electrons sort of jump down this gradient of 1.25 volts down to oxygen, which is here at plus 0.82. And this energy you get by moving charge across this potential can be released to do work, and that work is pumping protons across the membrane. So that's where the proton-motive force comes from. That's what it is. Now let's talk about how we want to sense it. And this slide is really why I'm here today. You know, we were interested in sort of developing these sensors, but we realized that these PMF-dependent processes, you really need a good way to measure the proton-motive force on fast timescales, and there really wasn't a good way to do that for a while. So there are voltage-sensitive membrane dyes which work very quickly, but unfortunately they don't work in E. coli because E. coli's membrane structure is such that the dyes don't bind very well to it. So these were out of the question for us. Patch clamping, where you take a small segment of membrane and attach an electrode to it to measure membrane potential, works really well in neuroscience when you have neurons. The problem is in E. coli, the membrane size is so small that you actually can't get a good amount of membrane to do patch clamping. So that was unfortunately out of the question as well. There are other molecular probes that have been around since the 80s, but these measurements only work on bulk cell samples, so you can't get individual cell membrane potential over time. And also, your measurements are averaged over minutes and long time periods, so these were out of the question as well. But thankfully, about 3 years ago, a group at Harvard University, Adam Cohen's lab, found this protein in nature called proteorhodopsin that takes in light and then uses that energy to pump protons across the membrane. And they did a clever mutation which made the protein work in reverse, where if it's attached to a proton, the protein becomes fluorescent. It gives off light. So now we have a proton sensor that's optical, with an optical readout. And the really nice thing is that the proton-motive force actually changes the fluorescence profile. So when the proton-motive force is weak, this protein fluoresces more. And when it's strong, it fluoresces less. So this was our sensor. This is what we decided to use to start probing the process. And the nice thing is we have the readout on the order of 100 milliseconds with a sensitive camera. So and it's great because the cells make it. It's biocompatible because it's a protein they can just stick in their membranes. And so we were off to the races to start measuring the proton-motive force. Okay. So it's time to talk about how we applied the sensor. So one example is what we did as sort of a sanity check was just to do something that disrupts the membrane potential completely, that we know the PMF is completely dissipated, and just see how the sensor responds. So this is called an uncoupler. That's the lingo of these membrane physics, called FCCP. And what it does is it's a nonpolar molecule that can bind to protons and then diffuse across the membrane. So the cell makes all this effort to separate charge, but then this ionophore can grab the protons and sort of equalize and eventually destroy any gradient that you get across the membrane. So this was a video of what happens after we add FCCP, so I'll go ahead and play it. So there was a cell here that would turn bright. It would sort of go to the solid color like this cell was here. But we have this sort of fluorescence trace that gives you an idea of what actually happens. You would see the cell sort of slowly get brighter and brighter until it reached sort of a steady state value after all the diffusion of the FCCP was done, and it holds a constant fluorescence when its membrane potential is totally dissipated. So these are the kind of signals that you can see using our sensor props. But we were kind of interested in, what happens to PMF generation as we get rid of oxygen, as we get rid of our electron sink for our metabolism. So when you have a lot of oxygen, you can get a lot of metabolic throughput, where you have many electrons flowing from glucose to oxygen, and you can get fast generation of PMF. The cell makes a lot of energy. However, as that oxygen gets depleted, the actual kinetics of this reaction slow down, and you generate PMF more slowly. Okay, so we were just interested in what happens to the PMF signal during the transition between these states as the oxygen concentration decreases. So in order to do this, we made a setup that allowed us to flow nitrogen through the cells and allowed us to monitor them on an inverted fluorescence microscope as the cells used up all the oxygen in their environment. And this is what happens. So at 5 minutes, we flowed nitrogen in, and 25 minutes, we stopped the flow of nitrogen. So we just made sure the entire chamber was filled with nitrogen and just let the cells kind of keep metabolizing their glucose until they used up all the oxygen. Now, what you're looking at here is every 50 seconds, we average the fluorescence across all the cells in the field of view. There are about 100 in this particular one. And as you can see, they keep a relatively steady fluorescence rate. And as the nitrogen fills their environment and they're using all the oxygen, their fluorescence jumps up just a little bit to show that there's this transition to when they start doing more anaerobic type metabolism, and their proton-motive force generation is slower. And it's actually well known from these studies done in the 80s that the proton-motive force decreases now when there's no oxygen. So good, we were able to see sort of this jump up in fluorescence telling us that the cells on average are not making as much PMF. However, if you take the signal and look at it on a very quick time scale, you start to see something very interesting emerge. And so this is one trace from a single cell in the presence of oxygen. And you can see these little peaks start to show up. And these peaks only arise when there's a lot of oxygen around. And it's very interesting because these peaks represent transient depolarization of the membrane, where the PMF decreases maybe like 50 millivolts. And these pulses last for about 1 to 5 seconds. And it's not really clear why the cells are sort of putting all this energy into making PMF and then using it all up in these big, very rapidly. And what becomes very interesting is that, so since these pulses are rather big signals, we can do wavelet denoising that allows us to remove a lot of the high frequency noise and still keep the peaks and start doing analysis on them. So I want to show you two traces. One, these are both denoised. One from an oxygenated case, and one after the cells used up the oxygen after 30 minutes, and we measured them as well. And you start to see that it looked like there were a lot more pulses when there was the presence of oxygen than when most of the oxygen had been depleted. So we're kind of interested in why this might arise. But we wanted to make sure of ourselves. So if you zoom in on this trace, there are kind of two parameters we saw that might be interesting. One is the width of the pulse, which is really just the full width half maximum of the fluorescent spike. That's the amount of time the membrane is depolarized. Then we also were interested in the interpulse interval, so how rapidly the cells are spiking. And when we looked at these two parameters, we saw a very interesting difference between the two situations. And that's that cells that we recorded under oxygen, we got really strong clustering where the mean pulse width was only about a second. So the depolarized state was very short. And also the mean interpulse interval was on the order of 10 to 20 seconds. The cells were really spiking. So when they're generating a lot of energy, their membrane potential actually fluctuates a lot more, which is odd because it had been understood that cells like to maintain a constant membrane potential. But when the cells are under nitrogen and their metabolism is running more slowly, it actually looks like the cells, they don't do these large pulses anymore, where their membrane potential depolarizes quite so often. And they also stretch out a little bit in time, so the pulses get wider and wider. So one way to think about this, and we speculate that it's because when cells have oxygen, they're producing so much energy that they need a sort of relief valve. And so they have all sorts of processes that consume proton-motive force in some ion channels that can open up to relieve the pressure. And then they can, since they're generating PMF very quickly, they recover very fast, and so the pulses are very, very narrow. However, when the metabolic throughput decreases under nitrogen, the cells are producing energy much more slowly, and so their recovery from these low PMF events becomes a little more protracted, but also they don't have to sort of relieve the PMF quite so often. So it's an interesting dynamic story about the proton-motive force that we saw with the sensor. So in summary, we wanted to start understanding more internal processes in order to use biological cells as front ends. The proton-motive force is tied to a lot of internal processes, so that was our target. We used an optically sensitive, or an optical readout for the proton-motive force. And we started to pave the way for really understanding this signal and how it relates to metabolism. And it allows us to start playing with the cells now in different sensing techniques and see how we can start interfacing with the cells. So thank you.